Polymerase chain reaction. PCR is a technique used in the lab to make millions of copies of particular section of DNA. It was first developed in the 1980s. What is PCR? The polymerase chain reaction (PCR) was originally developed in the 1983 by the American biochemist Gary Mullis. He was awarded the Nobel Prize in Chemistry in 1993 for his pioneering work. PCR is used in molecular biology to make many copies or amplify small section of DNA or a gene. Using PCR, it is possible to generate thousands to millions of copies of particular section of DNA from a very small amount of DNA. PCR is a common tool used in medical biology research labs. It is used in the early stage of processing DNA for sequencing, for detecting the presence or absence of a gene to help identify pathogens during infections, and when generating forensic DNA profiles from tiny samples of DNA. How does PCR work? The principle behind every PCR, whatever the sample of DNA, are the same. 5 core ingredients are required to set up a PCR. We will explain exactly what each of these do as we go along. These are the DNA template to be copied, primers, short stretches of DNA that initiate the PCR reaction designed to bind to either side of the section of DNA you want to copy. DNA nucleotide bases, also known as DNTPs. DNA bases A, C, G, and T are the building blocks of DNA and are needed to construct the new strand of DNA. Tag polymerase enzyme to add in the new DNA bases and buffer to ensure the right conditions for the reaction. PCR involves of heating and cooling, called thermocycling, which is carried out by the machine. There are 3 main stages: denaturing and annealing and extending. Denaturing, when the double-stranded template DNA is heated to separate it into 2 single strands. Annealing, when the temperature is lowered to enable the DNA primers to attach to the template DNA. And extending, when the temperature is raised and the new strand of DNA is made by the tag polymerase enzyme. These 3 stages are repeated 20 to 40 times, doubling the number of DNA copies each time. First, what happens at each stage of PCR? Denaturing stage. During this stage, the cocktail containing the template DNA and all the other core ingredients is heated to 94-95 degrees Celsius. This results in 2 single strands of DNA, which will act as templates for the production of the new strands of DNA. This usually takes between 15 to 30 seconds. Annealing stage. During this stage, the reaction is cooled to 15 to 65 degrees Celsius. This enables the primers to attach to a specific location on the single-stranded template DNA by way of the hydrogen bonding. The exact temperature depends on the melting temperature of the primers you are using. Primers are single-stranded of DNA or RNA sequence that are around 20 to 30 bases in length. The primers are designed to be complementary in sequence to short sections of DNA on each end of the sequence to be copied. The 2 separated strands of DNA are complementary and run in opposite directions. From one end, the 5 end, to the other, the 3 end. As a result, there are 2 primers, a forward primer and a reverse primer. This step usually takes about 10 to 30 seconds. Extending stage. During this final step, the heat is increased to 72 degrees Celsius to enable the new DNA to be made by a special tag DNA polymerase enzyme, which has DNA bases. Tag DNA polymerase is an enzyme taken from the heat-loving bacteria Thermus aquaticus. This bacteria normally lives in hot springs, so can tolerate temperatures above 80 degrees Celsius. 72 is the optimum temperature for the tag polymerase to build the complementary strand. It attaches to the primer and then adds DNA bases to the single strand one by one in the 5 to 3 direction. The result is a brand new strand of DNA and a double-stranded molecule of DNA. The duration of this step depends on the length of DNA sequence being amplified, but usually takes around 1 minute to copy 1000 DNA bases. These 3 processes of thermocycling is repeated 20 to 40 times to produce lots of copies of the DNA sequence of interest. The new fragments of DNA that are made during PCR also serve as templates to which the DNA polymerase enzyme can attach and start make DNA. The result is a huge number of copies of the specific DNA segments produced in a relatively short period of time. This illustration shows how the polymerase chain reaction PCR produces lots of copies of DNA. Thanks for listening. Project made by Lana Al-Wazour.